Thank you, everybody, for joining us back here at Hemostemix's channel. We have another special guest for you all today. Today, we are talking to Dr. Nadia Giannetti. We're going to dive into her knowledge in relations to stem cells. But Nadia, how are you doing?
I'm very well, thank you, Michael. How are you?
I'm doing really well, and I'm really excited to get to know a bit about your knowledge and glean some insight from you. But before we do, talk to me about your relationship to Thomas and Hemostemix.
I met Thomas and Hemostemix. They came to me, I want to say about 6 months ago. It was after we had a feature in the news about some work we're doing with stem cell therapy, a clinical study called Heart in a Dish, whereby we are, creating stem cells from patients with dilated cardiomyopathy, using those stem cells, creating cardiomyocytes, and trying to understand why the cardiomyocytes are functioning abnormally. So it's a different kind of line of work than what Hemostemix is doing, but it's in the stem cell world. And so they contacted myself and my colleagues to tell us about what they were doing and what they were interested in doing.
Wonderful. Yeah. I mean, stem cells are kind of they're a groundbreaking thing, and there's a lot of promise still wrapped into them. And as I understand it, since 2021, you are the associate physician in chief of the Department of Medicine at the McGill University Health Center. And prior to that, since 2010, you were the chief of cardiology.
That's correct.
Wow. Yeah, that's some pretty big accolades there. So, like, how much of your time do you practice medicine seeing patients with ischemic cardiomyopathy?
So really, it's patients with ischemic cardiomyopathy or heart failure. That's by far my primary clinical what I do clinically. So that's I
Mhm.
almost do only that clinically.
Okay.
So that I take care of patients with heart failure. Sometimes it's milder, but sometimes it's very severe. So what I really specialize in is the what we call advanced heart failure. So people for whom options are running out, people who
Mhm.
have exhausted most medical therapy that's available, any kind of device therapy, valvular therapy, and now they're looking for other solutions. In rare cases, people are candidates for transplant, and we put them on the transplant list, and they wait. Could be years.
Mhm.
Or sometimes patients need to go on to what we call mechanical circulatory supports or LVADs to get them to transplant or to live out, the remainder of their days. So that's really the population I take care of, and there are great medical therapies, and most people get better with medical therapy, but some don't.
Mhm.
And so what we're looking for, always looking for new solutions, new options to offer patients who have heart failure, and live a poor quality of life. And so we're always looking for solutions, and that's kind of what makes hemostemics interesting.
Yeah, that makes sense. And could you kind of like help us understand a bit, like what are these people who have ischemic cardiomyopathy experience? Can you give us a sense of the things they're going through?
Yeah. So generally speaking, the symptoms of ischemic cardiomyopathy, or which is basically symptoms of heart failure, are really shortness of breath, low effort endurance, fatigue, swelling in their legs, sometimes in their abdomen. Uh, Sometimes they have chest pain as well. They can't lie down flat. They're short of breath, even just lying down. In extreme cases, the quality of life is extremely poor, and people can't walk, 10 steps without being short of breath. So,
Mhm.
it's quite debilitating, not to
Mhm.
mention the hospitalization. So patients with heart failure have frequent hospitalizations because they become congested, they retain fluid, and they're hospitalized to get rid of that fluid.
Yeah, yeah, definitely does affect their life. And as you had said, that's why you're here looking for solutions in other ways. So, turning to that a little bit more, how would you rate the ACP1 animal study of ischemic cardiomyopathy? I'm going to say that word 100 times. I'm going to mess it
Yeah,
up sometimes. Completed by your colleagues at the University of Toronto, that is published in the European Journal of Heart Failure.
yeah. So I really like that study a lot. First of all, it was done in a great center. I mean,
Mhm.
Toronto is one of our another great Canadian site, a very credible, very high level research institute at University of Toronto. This was done in rats where they ligated or basically, occluded the main artery of the rat to create a heart attack. So obviously
Mhm.
it's an animal, it's an artificial model because it's in an animal, but this is what you see when people have, it simulates a heart attack in the main artery of a heart. of the heart, and that leaves a residual deficit. So an area, a scar in the heart, where the heart doesn't beat adequately, and there's what
Mhm.
we call a reduced ejection fraction. So that means the pumping ability is reduced. And so it simulates a model of what we see classically in patients who come in with heart attacks. And what they did is, they had a control group and they had a group that was treated, and there was a group that was treated given stem cells directly in the muscle, and another group that was treated given stem cells through the blood vessels,
Mhm.
which are two different approaches. Both groups fared better than the control group, so it was actually, really nice, a nice result with both group. A little bit better in the group with the intramuscular, so the direct injection into the muscle,
Mhm.
compared to the group that had the stem cell, the ACPs injected into the vasculature. But overall, really, actually good results in both groups. You're always compared to control, so that's very important to have a control arm, and so I think, for me, those are very favorable results. It's a well-designed study. Because it's in animals, obviously you have access to heart tissue, so you can see what happens to the scar, you can see what happens to the muscle, and so you can see that the stem cells are really going into the muscle and improving the muscle function. So it's actually a very promising study done in animals.
Yeah, yeah, it sounds like they both did pretty well. So
Yeah,
I guess from there, like how significant would you say this is that they was able to see new blood vessel formation and
yeah.
density?
I think it's very significant in that, the way I look at it is, again, we're talking about people who've exhausted standard medical therapy. The medical
Mhm.
therapy in patients with heart failure with reduced ejection fraction, patients like this, it's a great cocktail that we follow. Everyone follows the same cocktails, whether you're looking at the Canadian, the American, the European guidelines, okay? But you get to a point whereby you've exhausted that cocktail. And so anything that you can do on top of that is a benefit. So, in rats, you're looking at an improved ejection fraction. Even if it's 4% or 5%, that is a good thing. So, at a very low risk. So anything you can do to improve the heart function at a reduced risk is welcome. Does it change? Does it make, is it the equivalent of giving a brand new heart? It's not. Okay?
Mhm.
You're not giving a brand new heart. We're not talking about normalizing the heart function. We're talking about people who've exhausted all conventional therapies with medications and device and valves, whatever you can offer them, and now saying, okay, you're here now. There's nothing I have left to offer you. Can I continue to make you better?
Mhm.
I like the concept of ACPs because it is, what we see in the literature. Because aside from the animal studies, there are human studies. These are
Mhm.
not randomized controlled trials, but they do show safety, that these were injected in humans safely. And if you could imagine a scenario where inject these cells in humans, and it's safe and they improve the heart function, even if it's a little bit at a time,
Mhm.
no one says you only have to do it. We can only do it once. Okay.
True.
You
Yeah.
can imagine a scenario where you do it now, and maybe in a year you go back and you do it again, and another year. So we don't have that data. I'm not pretending that I know what that's going to show.
Mhm.
We don't have that data to tell us that maybe you could do this every year. But I think that is something that merits further investigation, and I think the possibilities are great. I think the drawbacks are very low because all you're doing is taking peripheral blood and injecting, patient's own blood. This is what we call, autologous. You're not giving people foreign materials, no foreign tissue.
Yeah.
There isn't a risk of rejection, and in a well-controlled environment, it appears to be very safe. Again,
Mhm.
we have to go through rigorous testing, but everything suggests it's very safe. So anything that can help. I mean, most of these patients will tell you whatever you can do to help me, they're willing to try. Okay,
Yeah.
so I look at this very favorably, wanting to help my patients and wanting to improve their quality of life.
Well, yeah, that's good that so far things have came up in terms of there hasn't been any issues. It seems safe up to this
Yeah,
point, as you said, at least as far as we know. And it apparently also had a statistically significant impact on angina, which is like chest
yeah,
pain.
Yeah,
Can you talk to me a bit more about that? Because pain is a huge damper on the quality of life with people. It
yeah,
can often lead to doing other things to try to get rid of said pain, like turning to medications and so on, which
yeah.
can go down a whole plethora of other issues.
So, we have patients, it's not necessarily exactly the same population. Sometimes they are ischemic cardiomyopathy, sometimes they're not. Sometimes they're patients who have a normal ejection fraction, don't have heart failure, but what they have is diffuse coronary disease, small vessels that is not amenable to your standard angioplasty
Hmm.
or your CTO opening or your bypass, anything that we do in standard in a hospital, in a cath lab, or in the surgical in the operating room. Some people have, no matter what you do, despite all the medications you give and the procedures you do, have intractable or very low threshold angina. Okay,
Hmm.
so it's the same idea. If you can rebuild some of their vessels, you can further develop their vessels and reduce their angina. That could, once again, really help patients in terms of their quality of life, right? So again, very exciting.
Yeah. And turning to things that we can statistically measure, can you help us understand like the significance for a patient that can improve their walking distance by say 24% in like the 6 minute walk test? Is this like a serious improvement?
Yeah, it is. It is. Quality of life for patients is huge. So, you have, it's always a risk. There's always a risk benefit. Risk. You have to look at the the risk benefit ratio, and walking 24% more. If you talk to someone who can't walk more than 10 meters, well, 24% more is still 24%
Yeah.
more, right?
True, yeah.
So, if you weigh, you have to weigh obviously the cost, how effective things are, what the risks are that are involved. So you have to weigh all of that. But these patients would tell you that they're going to do anything to get better. You know, anything to avoid, getting their heart taken out of their body and replaced with someone else's heart, and with
True.
all the immunosuppression that goes with it, right? Like, which is extreme when you think about what the treatment that we offer patients with end-stage heart failure is to literally take out their heart and replace it with another human being's heart,
Yeah.
and give them medications the rest of their life to prevent rejection. So medication that suppresses the immune system, that is pretty extreme. Okay,
Mhm.
and that will come when there's a donor available. It's not on the shelf. You can't do that. Just because you decide that you want it now, you won't necessarily get it now. So we're talking about, treatment options that are quite extreme, such as heart transplant. So if you can make people better and avoid a transplant, or better even while they're waiting for transplant, these are the type of things that are very welcome
Yeah.
at a reasonable cost and at a reasonable risk, obviously.
Yeah, fair enough. The risk seems minimally invasive versus all the other options. And as you had said, not to mention that those other options are not things that are always readily available.
That's right.
And obviously, like we don't have we're not quite to that point where we can just make as many hearts as we need right now.
That's
And
right.
it has to come from a real human being. So it's not something we have supply chains sorted out for at all times. But kind of from there, so in this study of the 41 patients who received direct injection of ACP by Dr. Kitt, it had demonstrated statistically significant 4.8 improvement of the ejection fraction. Can you like talk to me how unique that percentage of improvement is in these populations?
Yeah. So, listen, it's a good question. Many of the medications that we use, whether they're beta blockers or ACE inhibitors, or ivabradine or spironolactone, that's what they do. Okay, they may have other effects. They're medications, and they have
Mhm.
other effects. But if you look at how much they improve the ejection fraction, that's basically what they're going to do. Okay, each individual drug might increase your ejection fraction from 3%, by 3 or 6% absolute.
Mhm.
The best that we have on the market that we've seen with the Cubitril valsartan, maybe out to 12 months, maybe up to 9% improvement in ejection fraction.
Mhm.
But you're talking about medications here that have side effects. Lower your blood pressure, can affect your kidney function, can affect your potassium, that are sort of systemic, right? Because they're medications, so they have other effects. But if you look at what they do to the ejection fraction, none of the medications that we use every day improve the ejection fraction much more than that. Okay,
Mhm.
And people do feel better. Now, again, medications are more complicated. They have potentially other effects. They're not just there to improve the ejection fraction. Okay, but if you look at, excuse me, the actual effect on the ejection fraction, 5%, 4.5 to 5% is pretty good. That's what you expect most of your medications in our cocktail is meant to do.
Okay. Yeah.
And we give those medications without hesitation. I mean, to improve the ejection fraction. Again, it becomes additive. You add 2-3 medications, if they each improve the ejection fraction in a little amount, and then you add this on top, you would not use ACPs in absence of anything else. Nobody's saying that they would replace medical therapy or replace anything else we're doing. Okay, we're talking about additive, adding to what we have in patients who remain very sick and symptomatic despite our best efforts.
Mm-hmm. Yeah, it does make sense. And as you had mentioned, everything else comes with it's essentially a double-edged sword in terms of the other issues. But at the end of the day, improving that ejection fraction percentage is a really big deal in giving people a quality life. Yeah. And there was another study done with 106 patients by Dr. Supachai using a balloon angioplasty technique. Also had a significant improvement of about 4.7%.
Yeah.
And it seems that like in that the ACP cells migrated to the site of ischemia and embed at the site of ischemia to regenerate it. Is that what you think is going on, hypothetically?
Yeah, I think, I mean, that's what we hope to see. Yes, I think that what you want to see is that this goes to the site where there is abnormal cell function and helps to replace or to enhance that cell function, the cell function that you see at that site. Mm-hmm. At the scar or at the periphery of the scar, that there's basically assistance to, muscle cells that are just not functioning adequately.
Makes sense. And the last study we'll talk about here is this retrospective study of 53 consecutive patients treated for ischemic cardiomyopathy. There I go again. Also has a statistically significant improvement in ejection fraction of 4.8%. So why is a phase 2 study of ACP directly injected into the heart of patients suffering from ischemic cardiomyopathy the logical next clinical trial here?
So the studies you've described, apart from the animal study at U of T, are studies that were done without control groups. Mm-hmm. Okay. And so one could argue that that some of it is a placebo effect, okay, or just an effect of being treated, or just an effect of time, okay. So the results are super interesting, and they're like very thought provoking, but they are not conclusive, okay. Mm-hmm. Until you do a proper blinded placebo controlled study, okay. So what is needed now, and what we are, my team at McGill is designing, a study of an intramyocardial injection of ACPs in patients with ischemic cardiomyopathy that will be blinded to the patient, blinded to the investigator, okay, and randomized controlled, okay. So where there's no potential selection bias of the patient who is or is not going to improve. Okay, so these studies that you mentioned, okay, apart from the rat study, they're not randomized controls. There's no control group, and the investigators are not blinded. So they're thought-provoking, they're hypothesis generating, they're interesting, but they are not conclusive. And to move forward with a large phase 3, multinational, multi-center, thousands of patient kind of trial, or, high hundreds kind of trial, we really need a phase 2 proper randomized controlled blinded study. And that's what we hope to do. We're looking at we've done what we call a power calculation. We need about 100 participants to do that. I am incredibly excited, okay, to be involved in this, to be able to offer this to my patients, to at least offer the trial of this. Okay, I'm not going to tell my patients that I know whether or not it works. I will tell my patients that there is data that suggests that this could help them, and we want to know definitively. And this phase 2 trial is going to give us that information. We will also look at any kind of adverse events will be measured, and we have a list of adverse events to look for, and we want to be able to show how this can be done safely. So we want to show really safety and efficacy in a randomized controlled blinded study.
Mm-hmm. Yeah, I mean, I really look forward to hearing more about that. In terms of timeline, like when can people expect to potentially hear updates from that, and where could people go to learn more about that
Yeah.
study?
So it's a good question. So I'm hoping that we'll get this off the ground by the end of 2023. Start to recruit. I hope that we can start to recruit our first participant. And where to get information on it, it'll be at the Hemostemix website. You should be able to see some of this information, and at our own McGill University Health Center Research Institute on our website as well. So yeah, I'm very, very excited to test this out. Really, I mean, it is a phase 2 trial. We're testing out something that looks promising.
Mm-hmm. All right. Well, Dr. Giannetti, thank you so much for coming on and sharing your insight on this. I really learned a lot today myself. Hope everybody watching did too. And if they have any questions, please don't be afraid to send them over to us. But for now, it was great meeting for you. Look forward to chatting again sometime. Thank you.
Thank you very much, Michael. Thank you for your time. Bye-bye.